Hi, my name is Elizabeth Warden. Here's the piece of paper. Elizabeth Warden. Today is October 14th, 2022. I'm going to go ahead and do my presentation on chemoreception. So, just a general overview. Chemoreception is the ability to sense chemical stimuli in the environment. This is primarily done through sensory input, and in this case we're talking about olfactory and taste components. It's one of the oldest senses in the natural world, from prehistoric simple single-celled organisms all the way up to humans, unlike other senses. Just an overview of olfaction, or the sense of smell. This is the pathway that it takes. So the olfactory system takes place in the nose. The chemical signals are received by the olfactory sensory neurons in the olfactory epithelium, which you can see here, which is deep within the nasal cavity. The olfactory sensory neuron axons form the cranial nerve 1, and so that goes up. It's not letting me see my mouse, but it goes up to meet the olfactory bulb, which is this blue thing here on the underneath side of the brain in this diagram. The cranial nerve 1, which is the axon, connects to the olfactory bulb. It connects to the secondary sensory neurons there. The olfactory bulb extends to the olfactory tract, which is just an extension of it, which leads to the olfactory cortex in the brain, which sends signals to the cerebral cortex and the limbic system of the brain to receive these stimuli from the chemical signals that are inhaled. So just more detail about the olfactory epithelium, that first exposed area that receives olfactory stimuli. The olfactory epithelium is located deep within the nasal passage, like I said. There's a single dendrite that extends to the mucus layer and extends out, which picks up odorant molecules at odorant receptors. The odorant molecules must be absorbed by the mucus in order to be received by the dendrite of the neuron. The Bowman's gland, which this diagram shows, is this tannish structure in the center. That's the Bowman's gland. It creates the mucus that the odorant molecules are absorbed into. That these blue structures are the neurons, the olfactory neurons. There's around 400 different olfactory receptor proteins in humans, which account for a very wide spectrum of smells, and they work in conjunction to give in a whole smell. So it's extremely diverse and complex. Due to very short lifespans of the neurons, there's a basal cell layer that contains stem cells to replace old olfactory sensory neurons, and that's this structure here. It is this pinkish structure with a blue dot in the center. It creates new sensory neurons, and here on this you can see this neuron's dendrite hasn't quite reached yet. On the left, that's because it has not finished growing. So they're constantly being replaced and refreshed. There's a single, there's a typo there. It's supposed to say axon. It's a single axon that extends up to reach the olfactory bulb. So this axon here is what forms the cranial nerve 1 that we referred to earlier, that reaches up to the bulb. The yellow area is the cranial nerve 1, which is an extension of these axons from these neurons to send the signals that ultimately reach the brain. So, moving on to taste, the other chemoreceptor that we're talking about. It's very closely linked to olfaction. Taste is deciphered by a much narrower set of sensations, however, where olfaction has over 400 in humans, even more in other species, many more. Taste has a set of 5 currently that are currently recognized: sour, sweet, bitter, salty, and umami. There are multiple types of taste receptors to sense different tastes. So type 1, we think, senses salty taste, and we'll get to that later. Type 2 senses umami, bitter, and sweet, and that's pretty well understood. And then type 3 senses sour. And then there's basal receptors, sometimes called type 4 receptors, that are precursors to these. They're precursors to these receptors. So the umami, bitter, and sweet tastes are sensed with G protein couple receptors, while salty and sour are sensed with ion channels. So they have different mechanisms. It's more complex than just a simplistic mechanism that they all work similarly on. So sour tastes are regulated by hydrogen ion concentration in sour foods, most likely. The understanding of sour and salty is still a bit muddled. Sour is a little bit more understood than salty. The hydrogen ions are thought to, the concentration is thought to propagate a release of serotonin, which then propagates action potentials later on. But that's not necessarily the case for salty. They think that sodium concentration does something similar as with sour, but they're not 100% certain on that. There's other possible tastes that may be chemically received that are still being researched for further understanding, such as fattiness, carbonation, spiciness, and thickness, with associated chemical components like carbonation that's inputted into drinks or beverages, or capsaicin and spiciness, but they're not officially recognized because they don't know the mechanisms in which they take place or have enough research on it yet. So the pathway for taste, roughly, because it's a pretty complex, differentiated system. So on the taste buds, which have lots of receptors on it, a taste scent is dissolved into the saliva and mucus, and then that's shown here at the top at the number 1 on this diagram. It is then these taste ligands are then interacted with specific receptor cells to send a signal, which is specified based on the type of taste ligand. So if you're talking about sweet, umami, and bitter, we understand this pretty well. It's a G protein couple receptor. If you're talking about sour and salty, we think that it has to do with hydrogen being inputted into an ion channel, or sodium, most likely. Which the more understood sweet, bitter, and umami is on the left, and then the ion channel is on the right. This results in a neurotransmitter or ATP release, depending on which kind. So the neurotransmitter would be the serotonin release for the sour and salty, most likely. And then for the ATP release would be referring to the sweet, umami, or bitter ligand interacting with calcium and then causing an ATP, causing ATP to be formed. So this signal transduction, which is the process of doing this, just the overview of this more complex process, leads to the primary sensory neuron, which is seen at the bottom here, firing in action potential or action potentials based on the strength of the signal, which then signals sensory information to the brain. So either through neurotransmitters or through ATP stimulating the neuron. That is how taste functions. So that's the presentation. This all was cited from the textbook, which is cited here. All of the images are from the textbook as well. Thank you.